Apart from this, the starting with 2 kingdom, 3 kingdom, 5 kingdom and 6 kingdom classifications, there are some other group of individuals are there. Their position was not being shown in any of the classifications that were given by different scientists. So those individuals are viruses, viroids and prions. Viruses are the organisms which are considered to be the connecting link between the living and the non-living organisms. So let us start with the first group. And before that, all these individuals, viruses, viroids and prions, all these are ultra microscopic, and they requires the electron microscope or the light microscope for their observation. Starting with the viruses, viruses are considered to be the acellular structures. Here they does not have any particular cellular organization, and these are considered to be the obligatory parasites. And that is without host they cannot survive. That means when they are out of the host they are considered as a non-living, and when they are present or attached to the host they are considered to be the living. That is the reason why they are acting as a connection link between living and the non-living organisms. Then these structures, so how can we say that these are not having cellular structure? How can we include them in living organisms? Means the only thing that they contains genetic material. The genetic material will be in the either in the form of DNA or RNA. The other thing is it can undergo mutation also. So, and whenever it is present inside the host organism, it can perform the metabolic functions. So because of all these characteristic features, we cannot say that this is a non-living organism. That is the reason why the viruses, despite of acellular nature, they have been considered as a living materials or living organisms. The history related to the viruses, initially some scientists they consider to be it as a fluid, but later they are considered to be the filterable agents. But later it have been confirmed that they are having the particle nature. For the first time, Stanley had crystallized the viruses, that is the tobacco mosaic virus, that is TMV virus. Likewise, many scientists are there, Ivanovsky, Bajerinski, like many scientists are there who done research work related to the history or initial discoveries of the viruses. So viruses are chemically called as a nucleoproteins. Why? Because they contain nucleic acid and protein part. The protein part is called as capsid. Generally, the nucleic acid will be present in the core region, the central part, and which is surrounded by the protein part, which is considered as a protective sheet or the capsid. And here the capsid is made up of subunits. The subunits of capsid is called as capsomers. Capsomers. But in some of the animal viruses, apart from this capsid, so this capsid is proteinaceous in its nature, but in some of the animal viruses, apart from this protective capsid, one more additional layer is present, which is made up of lipids and proteins, that is called as peplose. And the subunits present in the peplose are called as peplomers. Peplomers. So this type of organization is there, as basically they are made up of nucleic acid and capsid, we call them as a nucleoproteins. And which type of genetic material is present? Either they may contain DNA or RNA, any one type of nucleic acid is present. If they are having DNA, generally DNA is present in animal viruses and RNA is present in the plant viruses. And if it is DNA, generally it should be double stranded DNA, but there are some exceptional cases where the DNA is in the single stranded condition also. But basically I said that DNA is present in animal viruses and RNA is present in some plant viruses. But there are some plant viruses like cauliflower mosaic virus and Dahlia mosaic virus. These are the two plant viruses which contain double-stranded DNA as the nucleic acid. And in the same way, there are some animal viruses which are having RNA as the genetic material. Likewise, polio virus and flu virus. These are the one which contains single-stranded RNA as genetic material. And generally when the DNA is present, it should be double-stranded, but there are some examples like bacteriophages, that is bacteriophage 5X174 and bacteriophage M14 and coliphase S13. So these are the examples where single-stranded DNA is present as a genetic material. In the same way, there are some examples where double-stranded RNA is present in the form of a genetic material, like wound tumor virus, rice dwarf virus, maize dwarf virus. So these are some of the examples where single-stranded, double-stranded RNA is present. So one other is a Rio virus. So these are some of the exceptional cases where instead of a double-stranded DNA, single-stranded DNA is present. These are the examples where instead of single-stranded RNA, double-stranded RNA is present. Okay, here let us discuss the brief characteristic features of some of the viral particles. Here, each virus particle is called as virion, which contains both nucleic acid and protein part. And here, let us discuss the typical plant virus, that is a TMV, tobacco mosaic virus. TMV virus was first crystallized virus that was done by Stanley scientist. This TMV is rod shaped with a spherical symmetry. And here in the rod shaped structure, we can see the helical shaped nucleic acid which occupies the core region. Core means the central part of the virus. So this is the chain of structure which is formed by the RNA, single-stranded RNA, and around this one we can see the presence of capsomers, that is a protein part which is made up of capsomers and individual unit, and the entire protein part is called as a capsid. So likewise we can see the rod shaped structure. This is a central core which is made up of single stranded RNA molecule. Then this entire part is called as a capsid, and individual structure of the capsid is called as capsomer. And generally the number of capsomers which are present in one capsid may be differing. Here In TMV virus, 2130 capsomers are present in each capsid, and each capsomer is one polypeptide chain, and this, as we know that the polypeptide chain or the protein chain is made up of subunits called as amino acids. So each polypeptide chain contains 158 amino acids. So these are brief characteristic features related to the rod shaped typical plant virus, that is TMV virus. Next one is typical example is a bacteriophage. Bacteriophage is a virus which infect bacteria, and bacteriophage were first discovered by Stuart scientist, but the name bacteriophage was coined by D. Harrell or D. Harrell. Here bacteriophages are named as T-even bacteriophages, like T2, T4, T6. Likewise, even number is given to them. If you see the typical bacteriophage, which is having tadpole shape with a head and a tail organization. In the head region and tail region will show variation in its nature in the symmetry. So it is showing, so head is hexagonal with a icosahedral symmetry, that is hexagonal structure will be there. Tail will have spherical symmetry. Head is having a cubical and tail is having spherical symmetry. If we observe the structure of the tadpole shaped organization of the bacteriophage, so this is a head region. Head region contains a hexagonal plate like organization. So this hexagonal plate like organization which contains this many triangular shaped face like organizations, which is called as icosahedral, and this icosahedral structure is made up of a polypeptide chains, that is called as a capsomers or capsid part is present, and inside this head region double stranded DNA is present in the form of a genetic material, and below this one we can see a thin and elongated structure. This is called as a tail. The tail is having the spherical organization, and in between the tail and the head region a collar is present. The function of this collar is unknown, and at the base of this tail we can see one hexagonal plate. This is called as tail plate, and if you observe this tail sheath, this is called as a tail, and it is surrounded by a capsomer structure called as a tail sheath. This tail sheath is made up of capsid, and this capsid is made up of subunits called as capsomers, and here the capsomers are 144 are there, and here they are arranged in 24 circles of 6 each, or 24 spirals of 6 each, and in the middle of this tail the central core is empty, and the tail plate, which is hexagonal, which contains pin like structures at each corner. So total of 6 tail pins are present. At each corner one tail pin is present. From each corner one fiber like structure will be arising. This is called as a tail fiber. A total of 6 tail fibers are present. So likewise, this is the shape of a tadpole shape of a T-even bacteriophage, which contains a head region and the tail portion. So likewise, this type of organization is there, and these bacteriophages are concerned with transduction process during the sexual reproduction of bacteria, where they will transfer the genetic material from one bacteria to the other bacteria. With the help of these tail fibers, these tail fibers have specific nature that they will attach to the specific points on the cell wall of bacteria. And apart from that one, the bacteriophages have the ability to secrete an enzyme called as lysozyme, which is a cell wall digesting enzyme. It will digest the cell wall of bacteria, and through that that will penetrate this genetic material by forming a hole on the bacterial cell wall. And if you observe the double stranded DNA, it is very big, and it is about 1000 times to the height of actual bacteriophage size. So that is, but all that DNA is coiled and fixed into the small head region. So these are the characteristic features related to the typical bacteriophage that was discovered by twat scientist. Next one is viroids or viroids. These were discovered by T.O. Diner scientist. Viroids are the viral particles which are having only nucleic acid, and they lack protein part. This type of viral particles are called as a viroids, and as we know that out of the nucleic acid and protein part, the nucleic acid is the infectious one. So here the infectious nucleic acid is present in the viroids, which cause damage or harm to the other living organisms. And because of only nucleic acid, their molecular weight will be low. And the RNA is a genetic material which is free RNA. And viroids will cause diseases like citrus exocartosis and potato spindle tuber. So these are two viruses, citrus exocartosis virus and potato spindle tuber virus, which are made up of only nucleic acid. Those are the examples for viroids. And next one is prions. Prions are the viral particles which are having only protein part, and they lack nucleic acid. Actually here we consider that nucleic acid is infectious part of the virus, whereas here the nucleic acid is absent. In such cases, the protein part is infectious, which cause diseases in other living organisms, and they will cause mad cow disease in cows and scrappy disease in sheep. So these are the other characteristic features related to the special type of virus particles called as prions.